Hello biologists, this is Miss R and we are going to do the first review of Semester B. For Quiz 101 we're going to talk about DNA and RNA. We're going to talk about how DNA copies itself to make more DNA. We talked about this when we talked about mitosis. We're also going to talk about how DNA makes mRNA in the process of transcription. And then we're going to talk briefly about how mRNA codes for protein and the process of translation. Remember, DNA makes more of DNA. That's replication. That's making copies. When DNA makes RNA, or mRNA to be more exact, that's transcription. When DNA makes protein, that's translation. DNA is a little bit like Morse code. There's a series of dots and dashes that code for all the words in the English language. And DNA uses more code than Morse code. Morse code only has two things, dots and dashes. DNA is a little bit different. The code is in the chromosomes in the nucleus of a cell, and that code uses 4 different options. Hopefully you can remember what these 4 different options are from semester 1 when we studied nucleic acids. The code in a nucleic acid is in the form of 4 bases. In DNA, that's cytosine, guanine, adenine, and thymine. And hopefully from semester 1 you can remember that DNA is short for deoxyribonucleic acid. You don't have to be able to spell that, but you should be able to say it. Deoxyribonucleic acid. This is a short history of DNA. In the mid-1800s, Friedrich Michner first discovered acids in the nuclei in the cells of pus. Here's a picture of some pus in someone's eye. When you have pink eye, you get very pussy eyes. Then after Michner, there was Chargaff, who discovered that there was a one-to-one relationship between these 2 bases, adenine and thymine, and a one-to-one relationship between guanine and cytosine. Chargaff did not understand this whole structure. That would come later, but he did understand and help the folks who came after him because he knew that for every adenine there was 1 thymine, and for every guanine there was only 1 cytosine. There's a relationship of 1-to-1 for these 2 bases and a relationship of 1-to-1 for these 2 bases, as stated right here. That helped Rosalind Franklin and later Watson and Crick come up with the structure. Rosalind Franklin, she was the person who discovered DNA was a spiral strand. She was a radiographer. She took a picture of the crystal structure of DNA. Francis Crick and James Watson looked at her picture and came up with the double helix structure that we all know well today. Now we know that DNA is the master copy of an organism's information. It's the big instruction book of the cell. And I happen to have a big instruction book of my kitchen right here. This is how to cook everything. It has a whole bunch of recipes, 2,000 in fact. But that's not even as many as your DNA. So this has a lot of directions in how to make a lot of different great things. It also stays in your kitchen. DNA, which I have a model of here, also has recipes for a whole bunch of different proteins, and it stays in your nucleus. During mitosis, DNA is copied exactly and passed on to new cells. This process, where we make an exact copy of the entire instruction book, is called replication. When you make a new copy of DNA, that's replication. You make another whole book. But let's talk about the structure a little bit more before we try to copy it. The structure looks really complicated, but it isn't. You can simplify it and think about it as a ladder. And we can talk about the sides of the ladder or the backbone of DNA, and then we can talk about the steps. The two different molecules that make up the sides or the handrails of the ladder are a sugar and a phosphate. This is deoxyribose, and this is a phosphate group. Here's a more close-up look. Here's the sugar. It's a pentagon shape. It's called a pentose sugar. Pent means 5. Hopefully you remember that from geometry. And the phosphate group right here glues the sugars together. It's like the linking Lego between two different sugar shapes. 4 different molecules make up the steps of the ladder. We talked about the 4 bases that are the code of DNA. And they follow Chargaff's rule. For every guanine, you have a cytosine. For every adenine, you have a thymine. See how there are 4 different colors to these steps? And orange always goes with blue, and pink always goes with green. That's G always pairs with C, and A always pairs with T in DNA. Now if you just twist the ladder, you've got your structure. Here's the ladder untwisted, and here's the ladder twisted. I took my ladder a bit apart at the top, and we'll talk more about that when we talk more in detail about replication in the next video. Remember that those steps are always found in pairs. G always pairs with C, and A always pairs with T. It's easy to remember how that works if you just remember GCAT. That way you remember the G goes with the C and the A goes with the T. The order of the nitrogen bases, whether there's ATC or CTA or TGC, that's the code that carries the information for the cell. So when DNA replicates itself, it has to make sure that code stays in the same order. So let's go back to our analogy of the cookbook. If we have DNA as the master recipe, what do you call it again when you make copies of the whole book? You basically do this. You take 1 copy of the book and you make 2 complete copies. Anybody remember? That's called translation. This process, making 2 complete copies of our DNA, our cookbook, and nothing leaves the nucleus, that's replication. Now, if you want to take the cookbook out of the kitchen, you want to kind of slim it down a little bit because you don't want to take this to the grocery store or your friend's house if you want to go make cookies at your friend's house. You want to scale this down a little bit and have something that's a little bit more workable. So you might copy one of the recipes onto a recipe card. You can make a whole bunch of these. If you spill stuff on it, it's no big deal. And that's kind of how your body treats mRNA. These can go to the nucleus, they can get used. You can make a whole bunch of them from 1 copy of DNA, and your body can use this recipe for a protein a whole bunch of times. And if it gets ruined, you can just make a new one from the master copy of your DNA. Now, when you write a recipe down, your handwriting is called script. This is a printing script right here. It's kind of like the font if you're word processing. So that's how I remember that the recipe card is transcription. Making DNA in the nucleus into mRNA is called transcription. Transcription has the word script in it. Now, if we want to take our recipe card and we want to take it into the kitchen and make something from it, like I've done right here, here's my recipe for chocolate double chocolate oatmeal cookies and I made a cookie out of it, that's where I translate the recipe. And this cookie here would be the protein. So the recipe is the mRNA, and the process of taking that recipe and making it into, using the instructions to code for something, using this code on my mRNA recipe to make a protein, that's translation. So transcription, then translation. So let's review a little bit. Proteins are made from the code in which molecule? We start with DNA, but that has to stay in the nucleus, so we can't use that to actually make proteins. So we have to write down something on the recipe card. We have to use a script to write something down. And that process is transcription. And here's your answer right here. RNA. Proteins are made from RNA. The original code comes from DNA, but you have to transcribe it into RNA so you can translate it into protein. RNA is the direct result of DNA. What do? You write it down, you use a script, which is a lot like a font, and when you're doing that, you're writing. So that's transcription. You can think of a medical transcriptionist as someone who listens to the tapes of doctors and writes down what they say. Or a legal transcriptionist does the same thing. You hear it and then you write it down. They're scribing or writing using a script. Sugar and phosphate make up what part of the DNA molecule? Remember, we talked about this is the sugar and this is the phosphate, and they link together. They're part of the backbone of DNA. So A would be the correct answer here. Sugar and phosphate would be part of the backbone. Now this asks you about Chargaff's rules. If one strand of DNA has 400 thiamine bases, how many adenine bases will the other strand have? Well, the abbreviation for thiamine is T, and the abbreviation for adenine is A. Let's look over here. If there's a T, there's an A. If there's an A, there's a T. It's 1 to 1. So if there are 400 of these, there are going to be 400 adenines. 400 thiamines, 400 T's mean 400 A's, or 400 adenines. So just for the big picture, if you're going DNA to DNA, that's replication. You make an exact copy of the entire molecule. If you go DNA to RNA, that's transcription. You're writing down the recipe on a recipe card using a script. You're scribing. If you're going DNA to protein, that's translation. You make something with the instructions that you've transcribed. And I'm wearing all these lovely beads because these are an example of the proteins you make. You take the individual amino acids and you link them together to make a protein. And we'll talk more about translation later in this unit. And then the protein will curl up looking something like this. So thanks for watching, and this is actually a picture of me. It's a perfect example of how one single cell with one copy of all of your DNA or 2 copies of all of your DNA, all your chromosomes, is translated into a person. You all came from one cell with just 2 copies of your DNA, and you turned into a person. Pretty amazing.